Hello everyone, welcome to Biotechnotebook. This video covers what is sterilization process and the methods of sterilization in bioprocessing. Sterilization process is used in bioprocessing to produce culture media, air and other processing materials, foods and pharmaceuticals which should be free from living organisms such as bacteria, fungi, viruses and their spores. Okay, so sterilization process used to kill all the foreign microorganisms which invade your fermentation process. In general, a fermentation product is produced by the culture of certain organisms or animal cell line in a nutrient medium. Okay, so a fermentation product is produced by culturing your microorganism, specific microorganism into your nutrient medium. This media contains your substrate which is converted into product by this specific microorganism or animal cell line. Okay, if a foreign microorganism, that is other than your specific microorganism, invades your fermentation process, then the following consequences may occur. The first one is the medium may support the growth of both production organism and the contaminant. Contaminant means a foreign organism. If medium supports the growth of both the organism, then foreign microorganism, that is your contaminant, may compete with the production organism for the nutrients. Okay, so it results in a loss of productivity. Okay, so it reduces the overall productivity of the fermentation process. Okay, then this contaminant may also produce some harmful byproducts. Okay, this byproduct may inhibit the growth of your production organism, which results in a loss of productivity. Then second one is outgrow of production organism. There are three types of fermentation process. One is batch fermentation, then continuous fermentation, then fed batch fermentation. Okay, if the fermentation is a continuous one, then your contaminant outgrow the production organism. That means your contaminant displace the production organism from the fermentation process. Okay, so you have only the contaminant inside your fermentation process, not your production organism. That is known as outgrow the production organism. Then third one is the foreign organism may contaminate the final product. That means you will get a impure final product. You won't get a pure final product. So it will reduce the quality of your product. The next one is the contaminant may produce some compounds which make subsequent extraction of the final product difficult. Okay, so after fermentation process there will be a extraction process. So using extraction process we will purify the final product. Okay, so if suppose your contaminant produce some compounds, we have to purify your product from these compounds also. So there will be a difficulty in your extraction process. Then this contaminant may degrade the desired product. If your contaminant degrade the desired product, then you can't use the product. Okay, so the nature of product will change. Next one is contamination of bacterial fermentation with bacteriophage, which results in the lysis of the culture. Okay, so in lytic cycle of bacteriophage, this bacteriophage enter into the bacterial cell. Then in bacterial cell which starts to replicate and it produces more progeny of bacteriophage. At the end of the process, this bacteriophage causes bacterial cell to burst or lyse and it releases its new phage particles. Okay, so in this process bacteriophage kills the bacterial cells, so thereby this phage reduces the number of bacterial cells present in the culture. Okay, so which leads to the decreased production of your desired products. So avoidance of contamination by foreign microorganisms may be achieved by using a pure inoculum to start the fermentation. So pure inoculum means pure culture. So this inoculum should contain a specific microorganism which you are going to use in your fermentation process. Then sterilizing the medium to be used. Okay, so before going to start the process you have to sterilize your medium. It should be free of all the foreign microorganisms, all the living organisms. Then you have to sterilize the fermenter vessel. Okay, so in fermenter vessel only you are going to produce your specific product. Then you have to sterilize all materials to be added to the fermentation during the process. Okay, whatever you are going to add in your fermenter, everything should be sterilized before it enters into the fermenter. Okay, then maintaining the aseptic conditions during the fermentation process. So during the fermentation process also you have to maintain your aseptic conditions. Okay, then let's see methods of sterilization. Okay, so first let's start with media. Media is nothing but your nutrient which is used to grow your specific microorganism in your fermentation process. Okay, so this media may be sterilized by different methods such as filtration, radiation, ultrasonic treatment, chemical treatment, then heat. Okay, so in these five methods, for practical reasons, steam is used almost universally. Steam is nothing but your moist heat. Okay, so heat is generally used for sterilization of your fermentation media. The major exception is filtration method. Okay, so this method is used to sterilize media for animal cell cultures because these media are completely soluble, and specifically these media contains heat labile components. Okay, so this filtration method is used to sterilize heat sensitive compound containing medias because high temperature can degrade or destroy these heat labile components. Okay, so filters with specific pore sizes may remove microorganisms from the liquid media. So here we are achieving the sterilization without heat. Next one is sterilization of your fermenter. Okay, so the fermenter, that is your fermenter vessel, should be so designed that it may be steam sterilized under pressure because before starting your fermentation process the fermenter should be free of all living organisms. Okay, steam sterilization is a method of using moist heat under pressure to kill all the microorganisms including spores. Okay, this sterilization process uses temperature at 121 degrees centigrade with pressure at 15 to 30 psi for approximately 20 minutes. So to kill the foreign microorganism, we are using high temperature and high pressure conditions. Okay, so this high temperature of 121 degree is achieved by using a saturated steam under pressure of 15 psi for 20 minutes. Then after your sterilization process is over, we have to barge a sterile air into the fermenter to maintain a positive pressure. Okay, or otherwise a vacuum may develop and unsterile air, that is impure air, is drawn into the vessel. This positive pressure means the pressure inside your fermenter vessel is higher than the outside the environment. Okay, then your medium may be sterilized in two methods. In the fermenter vessel itself you can sterilize medium. So this method is known as in situ sterilization of your media, or you can separately sterilize your media in a batch cooker or some vessels. Then you will add subsequently you will add that sterile media into your fermenter vessel. So that method is known as ex situ sterilization. So in situ method you will sterilize both the medium and the fermenter at the same time. Situ sterilization of media. So if the medium is sterilized in a separate batch cooker, then the fermenter has to be sterilized before this sterilized medium is added to it. Okay, as every point of entry to and exit from fermenter is a potential source of contamination. So each and every part of your fermentation vessel should be sterilized. Okay, so steams should be introduced through all the entry and exit points except the air outlet from which steam should be allowed to leave. Okay, so except air outlet point, remaining all the places, all the entry and exit points, you should sterilize the vessel. Then in fermenter, all pipes should be constructed as simply as possible and slope towards the drainage points to make sure that steam reaches all parts of the equipment. Next one is sterilization of the feeds. Okay, so a variety of additives may be administered to a fermentation during the process. Okay, so these additives includes nutrients, antifoaming agents, pH regulators, filterites and clarifying agents to improve your fermentation process. Okay, so it is essential that these materials should be in sterile condition. Then the sterilization of these additives depends upon the nature of additives and volume and feed rate at which it is administered. In continuous sterilization, you may add large quantity of your additives. So we have to consider the volume of additives to select your sterilization method. Then batch sterilization of feed liquids involve steam injection, held and storage vessels. The next one is sterilization of feed port. Okay, so addition of nutrients, acid or alkali to a small fermenter or normally made by silicon tube. So through silicon tube you used to add your additives, that is your nutrients or acid or alkali to a small fermenter. These silicon tubes are autoclaved separately and you will use to add your additives. Okay, then these additives are pumped by peristaltic pump after aseptic connection. Okay, then in large fermenter the nutrient reservoirs and associating pipings are usually an integrated part of your fermenter vessel. So with your vessel you will sterilize all these nutrient reservoir and associated pipes. Okay, there is no separate process in large fermenters. The next one is sterilization of the air supply. Sterile air will be required in very large volumes in many aerobic fermentation process. So aerobic fermentation process means your microorganism need oxygen to grow. Okay, so anaerobic means absence of oxygen, your microorganism will grow. So for aerobic fermentation process, you need very large volumes of air and it should be in sterile form. Okay, although there are number of ways of sterilizing air, only two have found permanent application. Okay, so these methods are heat sterilization, that is steam sterilization, and filtration as like media sterilization. For air sterilization also we are using these two methods, heat and filtration methods. So filtration method we will use glass wool, glass fiber, mineral slag wool and cartridge type filters. In association with the fermenter, these filters also should be sterilized before use. Okay, so each material you are using during your fermentation process should be sterilized. The next one is sterilization of sampling port. The sampling port is used to withdraw some amount of sample of your culture media for analysis. Okay, so sampling line and port should be sterilized by passing steam through the sampling line and port. Okay, we have to ensure all air is displaced by the steam. This maintain a sterile barrier between the fermenter and the outside environment. Next one is sterilization of the exhaust gas from the fermenter. Okay, we have to sterilize the exhaust gas. So exhaust gas also should be free of your microorganism which you use in your fermentation process. Okay, and any other contaminants also we have to eliminate it, because we are going to release this exhaust gas into the environment. Okay, so to protect our environment we have to sterilize the exhaust gas. So this can be achieved by using 0.2 micrometer filters on the outlet pipe. Under normal operation, aerosol formation may occur in the fermenter. Okay, so aerosols are nothing but suspensions of solids or liquid particles. So these liquid particles may moisturize your filter, or solid particles may plug into the filter. So frequently we have to check the condition of your filter. So to ensure a satisfactory operation, a cyclone separator for solids and a coalescer for liquids would be included upstream of two filters in series. Okay, so to separate solid particles we need a cyclone separator, and to remove liquid droplets you have to incorporate coalescer. Then these filters should be checked regularly to ensure that no viable cells are escaping into the environment. The next one is sterilization of liquid waste. Your liquid waste may contain harmful organism or some processed organism. So processed organism means which have been engineered to produce foreign products, and therefore certain heterologous genes are subject to strict contaminant regulations. Okay, so waste biomass of such organisms must be sterilized before disposal. Okay, so sterilization of your waste may be achieved by either batch or continuous sterilization. Batch sterilization involves sparging your steam into the holding tank. Whichever method you are using to sterilize your waste effluent, the effluent must be cooled to 60 degrees centigrade before it is discharged to waste. The sterilization regime used for destruction of processed organism will be different from that is used in sterilizing the medium. So the sterilization condition is different for both processed organism and for your medium. Next upcoming videos we will see in detail about batch sterilization, continuous sterilization and kinetics of sterilization. Then different filtration methods. Okay, thank you for watching my videos. If you like my videos, please do like, share and subscribe. Thank you.